Okay, so we have go through lipid metabolism yesterday. So today would be carbohydrate metabolism. All right, so these are the 5 pathways in carbohydrate metabolism. Okay, we have glycolysis, Krebs cycle, glycogenolysis, glycogenesis, and gluconeogenesis. Okay, so just to give you an overview, don't panic seeing these pathways, but all of these pathways are actually interrelated. Okay, we have glycolysis pathway over here. There are 16 steps. So glycolysis, glycogenolysis, glycogenesis, this is gluconeogenesis, and Krebs or TCA cycle. Okay, so the role of carbohydrate metabolism pathways are mainly to break down glucose and store glucose and synthesis glucose. Okay, so that's generally the 5 the things that these 5 pathways will achieve. Okay, so let's start with glycolysis. So glycolysis, what does that give idea to you? Glycolysis. Yes. So glyco is the glucose part, lysis is the breakdown. Okay. So in glycolysis, in short, it's from glucose, we are trying to turn them into pyruvate. Okay. So the initial product for glycolysis would be glucose that we obtain from our diet. Okay, after they have been digested by digestive enzyme like amylase, okay, for example, and the end product would be pyruvate. So this pyruvate is very important for energy production in our body. Okay, so pyruvate, in order for it to be formed from glucose, it requires aerobic condition. So what is aerobic condition? Yes, presence of oxygen. Okay, so in condition where there's lack of oxygen, okay, we call them as anaerobic condition. Glucose will produce lactate. Okay, so in our body normally, okay, everyone now is in aerobic state. Okay, so for example, you are doing intense exercise. For those of you who may have taken stairs instead of lifts just now, okay, your body might using up all the oxygen. Okay, most of the oxygen content. So your oxygen content may be low. So that is when lactate is produced, or you are more familiar with, for example, lactic acid. Okay, that's the thing that causing you muscle cramp after a very intense exercise. Okay, so another name for this glycolysis would be Embden-Meyerhof pathway. So sometimes in exams, they may ask you to explain the Embden-Meyerhof pathway. Okay, so don't be panicked, it's actually glycolysis pathway. Okay, so glycolysis is basically this pathway over here, starting from glucose down until pyruvate. So all of this takes place within the cytosol or inside the cell. Okay, then this pyruvate will be carried for, down here is the TCA cycle inside the mitochondria. Okay, so glycolysis alone, they have 2 steps or 2 stages, hexose stages and triose stage. Okay, so each of these steps, for example, from glucose to glucose 6-phosphate, there are specific enzymes that are catalyzing it. Okay. Okay, so another important thing that you need to understand, so in glycolysis, initial product will be glucose, end product will be pyruvate. Okay, so what are the other things that you have to understand? 1 molecule of glucose will yield 2 pyruvate. Okay, and this process, glycolysis, will release 2 ATP, meaning that glycolysis is where our body would need for them to acquire energy. Okay, so glycolysis is important to generate energy in our body. So starting from 1 glucose, ending 2 pyruvate and 2 ATP. Okay, and this process will yield water. Okay, and then site of reaction would be cytosol. Okay. So this is the step-by-step of glycolysis, starting from, actually this one I've divided into two sections. This is the first one until step 1 until 5. In the next slide would be step 6 until 6, 7, 8, 9, 10, 11. No. 6 until 10. Okay. So starting from glucose, this glucose will undergo reaction to form glucose 6-phosphate. Okay. I'm not sure how can I help you to remember this, but one of the way would be to understand the molecular changes. Okay. So from glucose, it will turn into glucose 6-phosphate, meaning that at the 6 carbon of glucose, there will be addition of phosphate group. So that's why it's glucose 6-phosphate, because at carbon 6, there's addition of phosphate group. Okay, so the enzyme is hexokinase, right? So hexokinase, it phosphorylate hexose. So this is the function. The name of enzyme is hexokinase. What hexokinase do? It phosphorylate hexose. It meaning that it's adding this phosphate group to the hexose carbon. Okay, so that is a bit of chemistry. Okay, so next is glucose 6-phosphate. It will be converted into fructose 6-phosphate. Okay, so they have the same chemical formula but different arrangement. So the enzyme that is catalyzing this reaction is isomerase. So remember, isomerase is about just changing the structure, but the chemical formula is the same. Okay, so it will convert glucose 6-phosphate into fructose 6-phosphate. So the enzyme would be isomerase. Okay, so next, fructose 6-phosphate will turn into fructose 1,6-phosphate, meaning that now there's another phosphate group added to it. So at carbon 1, we have one phosphate group, and at carbon 6, another phosphate group. So that is why fructose 1,6-phosphate. So another common enzyme that is adding phosphate group would be kinase. So apart from hexokinase, this one is phosphofructose kinase 1. Okay, so if you observe it carefully, phosphofructose kinase 1, meaning that it is adding phosphate to carbon 1. Okay, so that is why fructose 1,6-phosphate. Okay, so after fructose 1,6-phosphate, another reaction will take place, and that is glyceraldehyde 3-phosphate. Okay, so the reaction is catalyzed by aldolase. So aldolase, it will break aldehyde and alcohol group. So aldo is the aldehyde, the ol is the alcohol group. So this is what happened. Okay, let me zoom it a bit. Okay, so this is the structure of fructose 1,6-phosphate. Okay, so phosphate group at carbon 1 and phosphate group at carbon 6. Okay, so aldolase will break these two. Okay, turning into glyceraldehyde 3-phosphate. Okay, so moving on from this glyceraldehyde 3-phosphate, so now it will undergo another reaction catalyzed by glyceraldehyde 3-phosphate dehydrogenase, turning into 1,3-bisphosphoglycerate. Okay, So if you observe over here, so dehydrogenase, D means to remove, okay? Dehydrogen meaning it will remove hydrogen group. So if you observe over here, this hydrogen group is removed and a phosphate group is added. So glycerate, 3-phosphate dehydrogenase. So now this one turns into, they have 2 phosphate group at 1 and carbon 3. So it's 1,3 bis phospho, bis means 2 phosphoglycerate. Okay, so this 1,3 bis phosphoglycerate will undergo phosphoglycerate kinase. So kinase can do 2 things, either adding phosphate group or removing phosphate group. So in this case, this kinase will remove this phosphate group over here. So we no longer have this one, this 3 phosphate group. Sorry, the phosphate group at the carbon 3. So we only have 3 phosphoglycerate. Okay. So these 3 phosphoglycerate then undergo reaction. So if you observe, there's changes in this position. This phosphate group over here and this OH group here, they switch position. Okay, and that is catalyzed by phosphoglycerate mutase. So now this phosphate group is in the second carbon, so it's 2 phosphoglycerate. Okay, and this 2 phosphoglycerate will undergo reaction and form phosphoenolpyruvate. Okay, and this step is catalyzed by enolase. So removal of OH group over here. So we have phosphoenolpyruvate. And finally, a kinase will remove this phosphate group and form pyruvate. So this is the structure of pyruvate. Okay, so later on, take your time to memorize these steps and try to understand it with the aid of the structure changes, okay? So again, glycolysis start with, what are the initial? Yes, glucose, final product, pyruvate. So 1 glucose will yield 2 pyruvate and 2 ATP, okay? All right, so that's sufficient for now. So what's next? So what will happen to this pyruvate? So first, it will undergo alcoholic fermentation. So in some organism like yeast or acetic acid, so they will undergo alcoholic fermentation due to the anaerobic condition. Okay, so in alcohol fermentation, from pyruvate, they will yield 2 molecules of ethanol and also carbon dioxide. Okay, so this is the reaction. So pyruvate will undergo decarboxylation. Decarboxylation means it's removing this carboxyl group. So D means remove, like just now dehydrogen. So decarboxylation means to remove this carboxyl group. Okay, now it will turn into acetaldehyde and it will undergo reduction to form ethanol. So the key point for alcoholic fermentation is it occurs in yeast and acetic acid bacteria, and it will yield ethanol. Okay. I think that's the most important thing. Okay. So if you remember, if you have ever experienced making bread, you would put in yeast, or the commercial name would be What's the commercial name? When we make bread, kan kita letak yeast. What's the yeast commercial? No one ever made bread? Okay. What's the name? Tak apalah. Okay. So that will cause the bread to expand. So that expand is due to the carbon dioxide released by the yeast. Okay. Alright. So pyruvate can also undergo lactic acid fermentation. So if just now alcoholic fermentation is in yeast and acetic acid bacteria, for lactic acid fermentation, this can happen in the muscle tissue and also red blood cell. Okay, as I mentioned, this is during after intense exercise, for example, when the oxygen level in our body drop. Okay, so what catalyzes the formation of pyruvate into L-lactate is this lactate dehydrogenase. Okay, and this process is a reduction process. Okay, so I will give you some time to digest. 2 minutes to digest. So you can look back at this pathway and try to digest in 2 minutes. Those online, did you see the writings terbalik or you can read it?
Cannot read it. Too small.
Oh, too small, but it's not terbalik, eh? You cannot zoom lah for you.
No, it's the right one, but cannot see because it's too small.
Okay, I will tell you. Okay.
Thank you.
So throughout the topic for today, I want you to focus on this first before go into the detail for each pathway. Okay. So for example, just now we have go through glycolysis. So you can take note. Okay, what is the purpose while you digest? Okay, you can write on this. What is the purpose of that pathway? For example, glycolysis, breakdown of glucose. So that is the purpose. Okay. And then what is the precursor or starting point? So for glycolysis just now, it's glucose. And then what's the end product? So you can write in pyruvate. If you want to be more detailed, you can write in 1 glucose as the starting point and 2 pyruvate as the end point. Okay, does the pathway use ATP or produce ATP? So for glycolysis just now, it yields 2 ATP. Okay, so you can write in that. Okay, and then site of reaction. So just now, glycolysis happens in the cytosol. Okay. Okay, and then we can add on how many steps are there. So for glycolysis, it's 10 steps. Okay, so just every time that I give you time to digest today, you can go and summarize this. Okay, for each pathway. Okay. If you notice just now, the glycolysis ends at pyruvate, but this pyruvate can enter the mitochondria. Okay, so when pyruvate enter mitochondria, it will undergo a cycle that we call as TCA cycle or the Krebs cycle. So Krebs is the scientist. The name is actually tricarboxylic acid cycle or TCA cycle. Okay, so that is what we look into next. Okay. So this is the glycolysis part. So now glycolysis will yield pyruvate. So pyruvate will be converted into one precursor and enter the TCA cycle. Okay. So for TCA cycle, it happens in the mitochondria matrix, meaning inside the mitochondria. Okay, so another name would be, or the reaction would be oxidative decarboxylation. Okay, when we use the word oxidative, meaning that that reaction requires oxygen. So it has to happen in aerobic condition. That's why it happens in the mitochondria, because mitochondria has a lot of oxygen and oxidative molecule. Okay, so pyruvate will be transported into the mitochondria and pyruvate will be converted into acetyl-CoA. So who remember where can we find acetyl-CoA before this? In where? Lipid metabolism. So is it the precursor or end product? In lipid metabolism. Okay, short recap for you. You remember free fatty acid activated dapat apa? What do we get after activation? Acyl apa? Acyl-CoA, and then undergo go inside the mitochondria and form acetyl-CoA, and this acetyl-CoA will undergo oxidation process. Okay. So this happens inside the mitochondria itself. Okay. So this is the common point number 1, okay, for lipid and carbohydrate. So just now carbohydrate from glucose turns into pyruvate. So pyruvate will enter the mitochondria and form acetyl-CoA. So this happens in the mitochondria. Okay, so you cannot forget acetyl-CoA in biochemistry. Okay. Can someone take picture and send it in the group for those online? Thank you. Lama, I'm supposed to stop this now. Okay, okay, so this is how pyruvate turns into acetyl-CoA. Okay, there will be a release of NADH and also carbon dioxide. Okay, so why pyruvate is important in TCA cycle? Because the bonds between pyruvate molecules contain high energy. Okay, so cell require that energy. Okay, and that energy must be converted in the form of ATP. Okay, meaning that later on, when pyruvate turns into acetyl-CoA, it will release more ATP. Okay, and mitochondria will store that ATP. So if you remember back in chapter 2, one of the function of mitochondria is to produce ATP or synthesis ATP. So where does this ATP coming from? Pyruvate. Okay. So this Krebs cycle starts with the conversion of pyruvate into acetyl-CoA, and this acetyl-CoA will enter a cycle which we call as the citric acid cycle or TCA cycle or Krebs cycle. It's the same thing. Okay, you can ignore this activity. Later on we will do. Okay, so this TCA cycle, they have a lot of intermediates. Okay, from acetyl-CoA, they can turn into citrate, and then this citrate turn into isocitrate, and then alpha ketoglutarate, and so on. And these steps, similarly to glycolysis, have different enzymes. Okay, and each step will release different molecules. For example, NADH. So this step will release ATP. Okay. All right. So what are the importance of this TCA cycle to be converted to have these multiple steps? All right. So one of the importance will be this is what we call as intermediates. Okay. So another term that you need to be familiarized with is in this TCA cycle. Okay, citrate, isocitrate, alpha ketoglutarate, succinyl-CoA, succinate, fumarate, malate, and oxaloacetate. These are what we call as intermediates. So when I say intermediates in TCA cycle, so this is what I am referring to. Okay, so why TCA cycle have a lot of these intermediates? So because these intermediates have different functions. Okay, let me zoom this a bit. So these are the significance. For example, one of the intermediate, citrate. So this citrate is used to produce fatty acids in our body. Okay. So another example, alpha ketoglutarate. So alpha ketoglutarate can be undergo reactions and form glutamate. Okay, a type of amino acid which is important in protein, to build protein. Succinyl-CoA, they can form heme, one of the component in our blood, red blood cell, the hemoglobin. Okay, and so on. So that is the significance of TCA cycle having a lot of intermediates, because these intermediates are used as precursor in a variety of biosynthetic pathway. All right. So time to digest. So you can look back into the TCA cycle and try to match with this information. Okay. Those online, I will stop the Zoom now and enter back in 1 minute. Okay, because the time is 5 minutes left.
Sure, thank you.
Okay. If you have questions, please stop me, all right? Okay, if you actually Google this, you can find things like TCA cycle acronym. So people have been like creating acronym for you to remember the flow. You can use that, or you can create it by your own. Okay, you can take like Estelle, A, C, I, A, up to you. Okay, because usually every year the pattern will be in essay question. At least one of question will be coming from carbohydrate metabolism. So it can be glycolysis, it can be TCA cycle, or these pathways. So try to minimize losing marks because you don't remember the flow. So just to share with you, if you go in clinical setting, especially for those coming from biomedical science background, there's a lot of disease that are coming from defect from these pathways. So those taking clinical biochem later would still see this pathway, but you will see how, for example, the defect in this enzyme or the lack of this molecule can cause a certain disease. Okay, so for exam-wise, biochemistry is not fun at all, but learning-wise, knowledge-wise, application-wise, biochemistry has a lot of branch. So that's why in certain universities they are offering degree in biochemistry. Okay? They even have a specific degree for that because the field is so wide. All right. So we have covered glycolysis and then Krebs cycle. So Krebs cycle, just to for us to remember, is the follow-up or the continuation of glycolysis where pyruvate turns into acetyl-CoA, and that's when they yield ATP or energy. Okay, so next would be glycogenesis. Okay, so if lysis means to break down, what does genesis mean? Generate or synthesis. Okay, so glycogenesis means synthesis of glycogen. So if you remember, glycogen is the storage form of carbohydrate. So whenever there's excess glucose, our body will store them in the form of glycogen. Okay, so when do you think glycogenesis will take place? When will our body break down the storage? After meal? Yes. Sorry, I have to rephrase my question. Your answer is correct, but my question is wrong. When will our body produce glycogen? After meal, okay? Because after we have eaten, okay, now some part of the glucose is utilized for energy production, for example. So the excess one will be converted into glycogen. So that is when synthesis of glycogen takes place. Okay, so the main enzyme for this one would be, it's simple, glycogen synthase, because it will synthesis glycogen. Okay, glycogen synthase. So from glucose turn into glycogen. Okay. So for glycogenesis, to make it simple, just remember the purpose is to synthesis glycogen. Why? Because there's excess glucose. We need to store them. Okay. So the starting point is glucose. End point will be glycogen. Main enzyme will be glycogen synthase. Okay. For glycogenesis, let's stick to that. Okay. Okay. Next one would be glycogenolysis. So if glycogenesis is synthesis of glycogen, so glycogenolysis is the breakdown of glycogen. Okay. So when excess glucose, glycogenesis takes place. When does glycogenolysis takes place? If you don't eat when you are hungry, okay? Those online, okay? So quiet, you?
Yes.
Okay. All right, so, okay. So it's the conversion of glycogen into glucose. So it's the opposite of glycogenesis. So glycogenesis opposite is glycogenolysis. Okay. So now when our body is lack of glucose, so when after some time fasting or you after some time you are doing activity, glucose level drop. So the first reaction our body will do is to take back whatever that's in the storage. Okay, so that is glycogenolysis. Okay, so this one takes place in the muscle tissue and liver. Okay, because rapid storage. Okay, so meaning that storage just now happens also in muscle and liver, Whenever we need to retrieve them, the storage, it needs to happen at the same place. Okay, so triggered by low blood glucose, and the hormone involved would be glucagon. So after meal, insulin would be triggered, but in hungry state, glucagon will be triggered, okay, to kickstart glycogenolysis. Okay, so initial molecule glycogen, final molecule glucose, main enzyme glycogen phosphorylates. Okay, site of synthesis or site of reaction, muscle tissue and liver. Okay. Okay, so let's stick to that simple idea. Okay. Okay, time to digest. Okay, this is the extended notes that I've uploaded. Okay, the carbohydrate metabolism. So glycolysis, this is the written pathway. So the steps. Okay, and then the yield, and then the citric acid cycle, TCA cycle, the steps. So this is all the written form of it. And then biosynthesis of glycogen or glycogenesis. Okay, so the steps, the written form, so on. Okay, so you can refer to this if you need some guide to do the essay in the team, supplementary material. Okay, and towards the end, they have some disease or condition related to this pathway if there's defect or some damage. Okay, you can look into that. Okay, one more pathway for carbohydrate metabolism. So we have covered glycolysis, breakdown of glucose, and then pyruvate from glycolysis will enter Krebs cycle in the mitochondria, where it will generate energy and mitochondria will store the ATP. And then we have glycogenolysis, breakdown of glycogen store. We also have glycogenesis, when glucose is in excess, so we want to store that glucose. So now we have gluconeogenesis. Okay. So gluconeogenesis, if you ever forgot in exam, what are these things? Take some time to break down the name, okay? So gluconeogenesis. Gluco is of course related to glucose. Neo means new. Genesis means to generate or to synthesis. Okay? So synthesis of glucose. Okay. But now it's from non-carbohydrate source. Okay? So for example, if glycogenolysis before this, okay? Remember, glycogenolysis is when glycogen breakdowns into glucose. If that one is not sufficient to produce the glucose that our body needs, so our body will start to do glucogenesis. Okay? So remember, if glycogenolysis is not sufficient, then our body will start to do gluconeogenesis. Okay, so meaning that glycogen storage tak cukup, not sufficient. So we need glucose, but from other non-carbohydrate source. Okay, what are the sources? So they can produce glucose from lactate, pyruvate, glycerol, and glucogenic amino acids. Okay, from these intermediates of glycolysis itself, lactate and pyruvate, glycerol, the fats, and glucogenic amino acids, meaning the protein. So fats and protein can also eventually form glucose in gluconeogenesis. Okay. So it occurs mainly in the cytoplasm, in which cell? The liver cells, and small amount in the renal cortex within our kidney. Okay, that is a very small amount. Mainly it's in the cytoplasm of liver cells or in questions sometimes they may write it hepatocytes. So hepa is liver, so hepatocytes, liver cells. Okay. Okay, so in gluconeogenesis, the starting point is lactate, pyruvate, glycerol, glucogenic amino acid. These 4 things. That's the starting point. End point is glucose. Okay, and on top of glucose, we will have 4 ATP. Sorry. No. This gluconeogenesis will require 4 ATP, 2 GTP, and 2 NADH. Meaning that this gluconeogenesis require energy. It doesn't synthesize energy, it require energy. Okay, so in biochemistry term, gluconeogenesis is what we call as the expensive pathway because it will take away our ATP, okay, to produce glucose. But that is very important to maintain the glucose level in our body so that the organs will not collapse. Okay, so if you observe this diagram, it's actually overlapping between, there's an overlapping pathway. We have animals gluconeogenesis and plants gluconeogenesis, but animals and plants may share similar pathway. Okay, so plants, they don't have lactate or lactic acid. So gluconeogenesis from lactate only occurs in animals, while in plants they have carbon dioxide fixation. Okay, but the same pathway would be this from fats and also amino acids. Okay, so this amino acid, they will enter the TCA cycle and go back to pyruvate and eventually form glucose, similar to glycerol. Okay, meaning that it's over here. Okay, so this is the TCA cycle. So if you notice alpha ketoglutarate, they can form glutamate. Similarly, glutamate can reverse back and turn into alpha ketoglutarate. So glutamate is glucogenic amino acid, meaning that amino acid that can turn into glucose. Okay, so glutamate can turn back into alpha ketoglutarate and enter the TCA cycle. Okay, and this TCA cycle can reverse back, go back into acetyl-CoA and pyruvate. Okay, that's how from amino acid it reverse back to pyruvate and go up. Okay, this pathway of glycolysis, okay, and turn into glucose. So these are the examples of glucogenic amino acids. Okay, for example, what can form pyruvate is amino acid alanine, cysteine, glycine, and so on. Okay, what can form succinyl-CoA? Methionine, for example. So these bold wordings are all the intermediates in TCA cycle, and below them are the amino acids that can form them. Okay. All right, so that's carbohydrate metabolism. So what I want you to do is do this table. So it's basically this one, but please add in the ATP, use or synthesis of ATP, and also how many steps are there. Okay, and you don't need to upload this in Google Classroom. Complete this table and bring this in next class Tuesday. We will refer that for your tutorial. Okay.